good. I have a timer. I'll try not to go over too much so that everyone enjoys coffee. I will be discussing how we can, and if we can, interpret weight maps in terms of cognitive and clinical neuroscience. So I'll go a little quickly over this. I think Chris mentioned it quite a bit. So the model that we built, it is decision boundary, and it's characterized by a plane or hyperplane in the dimensionality of the data. It is characterized by a weight vector, and that weight vector can be expressed as a linear combination of the training examples, and it is used to make predictions for a new test example by taking the dot product between the weight vector and that test example. So we compute the weight vector by an optimization process where we try and fit the model with a data fit term where we denote the price that we pay when we make a mistake, and also with a regularization parameter, regularization term, that help us give some constraints on the weight vector. And actually that term will define how the weight map will look like. And it can be very different based on which regularization term you use to build your weight map. So as Chris mentioned, the weight vector can be plotted as, in this case, a brain image if you input brain images into your classifier. And so here it's the weight map of the HAXB dataset when you compare faces and houses. And you see those red spots and you kind of want to say, oh, well, probably most of my signal is there. So can we do that, and what exactly can we do with that map? So one thing to really keep in mind is that this is a predictive pattern. So we use this to predict labels. We do not try to really make an inference about whether a voxel carries signal about faces or houses. So these are two very different things, and we need to keep that in mind when we look at the weight maps. So if we make a prediction, we have a 4-dimensionality problem here with a weight map that has 4 voxels and 4 values, and we take a new example, and we predict and we obtain a function value of 1, and that's either the regression that you obtain, the regression target that you obtain, or it's the distance to the decision boundary. And one thing that is sometimes done is saying, well, I kind of don't care with those weights that have low values, so let's just take those out. And I'm going to just threshold to whatever has an amplitude that's lower, to 3. And if we do that, well, we do see that if we compute the prediction again, we obtain a different result. And so the truncated predictive function that we obtain does not correspond to the actual model we had before. And we see that even here, this is an illustration, but the value we obtain has actually even a different sign. So if this was a classification, it would be another class. So that's why basically when we say do not threshold the weight maps, it's because this is really a voting process, and every voxel, every feature you input, every weight has an importance. Another issue with weight maps is can be illustrated here. It was detailed in Schrouff et al. In 2014, and we can see here by illustrating with 2 voxels. So let's say in voxel 1 you have a signal that is a mixture of signal of interest and of noise. So you have signal of interest that correlates where your class is and noise that does not. And then in voxel 2 you have only noise. And well, the best model you could fit would actually be to take voxel 1 with a weight of 1 and voxel 2 with a weight of -1, so that you just retain that signal of interest here. And that's exactly what the classifier will do, because it's really helping it to make that decision between class 1 and class 2 here. So this is really to keep in mind as well, that the classifier will be using noise and actually try and take it out from the signal. So what can affect the magnitude of the weights? So a high coefficient or weight might be due to an association between the voxel signal and the labels, but also to other multivariate properties like correlated signal or correlated noise. Zero coefficient, on the other hand, might mean that there's no association, but also that it can be due to the type of regularization that you're using. So if you want to know if in a particular voxel there is signal about your labels, then doing an MVPA and looking at the weight map is probably not your best bet. And a GLM might be more efficient in that case. But what can we do with weight vectors? So we need to keep in mind it's the spatial representation of the predictive function. It shows the contribution of each feature to the prediction. It's multivariate, so we count threshold. And it's a mixture of signal of interest and of noise. And there are different techniques that have been developed over the years. This actually has gotten quite a lot of attention recently, so that list is getting longer and longer over the years that I give that talk. So I'll try and go quickly over it. But I think some approaches will be discussed further on in the session, so I might go quickly over some points. So there are different ways to deal, not to deal with it, but try to improve that. Can I go back? Okay, not too much. Okay, thank you. And they can intervene at different stages of the modeling. It can be a priori, so before you even try to fit your model. It can be during the model estimation at different stages, whether it's within the nested cross validation or outside of it. And it can also be a posteriori, once you have your weight map and you're just trying to look at it. And you're not affecting the model anymore at all. So if we start with a priori, what you can do is simply masking. So you choose a region of interest based on a priori knowledge, literature, or previous univariate results from an independent data set. And this is the result of the Haxby same data set, house versus faces, and it's using only the fuzzy-form gyrus. And what we're going to look at is not the weight map, actually, we just look at the accuracy. So it's really simple interpretation. Can I classify houses versus faces if I'm using only those regions of fusiform gyrus or not? And in that case, it's just like, is there a signal about my classes in there or not? So that's easier to look at than weight maps. And it's anatomically and functionally relevant, so it really answers in a hypothesis that you might have. But on the other hand, well, you need to have an hypothesis to be able to do that. You cannot threshold the obtained weight map, so the fusiform gyrus in that case is actually pretty big, but you cannot look at specific regions, subregions of that. It's an arbitrary choice of the number of regions and their sizes, and if you test it on multiple regions, you need to correct for multiple comparisons, whether that is using the permutation test that Carsten just mentioned or not. That technique can actually be extended to searchlight mapping. They're very related. And instead of choosing a region of interest based on literature, you just take a voxel and you take its neighbors as well. And you build a model based on that. And you replace the weight by an accuracy value for each voxel. So it's really a mapping approach where you get a map of accuracy instead of a weight map. And so it's easy to interpret, and you can threshold it when you run permutations and you test for the significance in each of the neighborhood. It is locally multivariate though, so if you are trying to look for a distributed pattern over the whole brain, this is not gonna help you. You also need to choose the radius of the neighborhood, which is an arbitrary parameter. You build multiple models, so it's, again, multiple comparisons, and it can be computationally expensive. So now we're getting, during model estimation, what we can do. And this is the first thing we can do, is do feature selection in a nested cross-validation. So we try and select a subset of informative features based on a criterion that is data-driven. There are different criterion that can be used. It can be a simple univariate t-test. It can be recursive feature elimination. So in that case, you start with your whole features, and then you take one out, and you see if each time you take one out, the accuracy improves or not. And then you select the subset with the highest accuracy, that leads to the highest accuracy within the inner loop of the cross-validation. And then you build your model and you test that on the validation set. So this technique is data-driven, so that is not the same as selecting a region of interest. And it identifies the most relevant voxels for prediction according to the criteria that you chose. Nested cross-validation can be expensive, but that limitation is to take with a grain of salt. It's more the criteria. If you choose different criteria, you might end up with very different subsets, I would say is more of a limitation. And then you can actually embed that feature selection process by using a regularization term that enforces sparsity or structured sparsity within your model. And there are a lot of different approaches. Probably the most well-known is the lasso by Tipshirani. And that gives you like very sparse, it's an L1 norm on the weight vector. And that gives you very sparse maps, actually. And regularization affects the weight map pretty heavily, although it can give very similar accuracies. So this figure was taken from Baldassare et al. In 2017, and subjects were reviewing pictures of pleasant and unpleasant images. And just the regularization was changed, and we see that we obtain very different sparsity and very different weight maps for pretty much the same accuracy. So the advantage of sparse algorithms is that they're data-driven, and it's embedded, the selection is embedded in the model, and it identifies a subset of predictive voxels that you can then look at, but you might have instability of the selected features based on which regularization term you choose. So now if we combine a little bit of a priori knowledge and of sparse algorithms, we can have atlas-based multiple kernel learning. And so with that type of model, you parcellate the brain according to an atlas. The choice of the atlas is yours. And here I chose the AL with 116 regions. For each region, we build a kernel, and the model is going to build a model for each region, and then it's going to combine those models into one final decision boundary. And those two processes of building a model per region and building a model that combines all those local boundaries, it's updating one another. So they're not like independent things. It's not the same as building a single model for each region. And you can enforce sparsity. And in that case, we chose to do it at the region level. So within each region, all the voxels will be used, but when we combine the regions, we're going to push some of the regions to have a weight, a contribution of 0. And so that gives you a sparse list of regions that contribute to your model. And we can see here, here we see the weight, so the contribution of each region, and this is the index, so there are 116 regions, and we see that this is very sparse. So there are only 13 regions that contribute to that model. And we can see the list, and we also see that, you know, they have names, and they are anatomically or functionally relevant based on which atlas you've chosen. So you can also look at these regions. And we see that Fuzzy form, for example, is high in the list, which is something that we would expect. And then we can go with inference form, and we can still look at the weights within that region, although that's not really helping us. And then one thing that I'm mentioning here because it's something that I've played with, and I've played especially with MKL. So I'm not mentioning it with other techniques, but it doesn't mean that it's not applying to other techniques. But I've played with intracranial EEG, and the signal to noise for intracranial EEG is actually pretty good. When you're looking at high-frequency broadband activity. And here you had a patient who was looking at different images of faces, buildings, and animals, and I compared that to a rest session. And I computed the MKL not using anatomically defined regions, but using each electrode separately. And we see that the MKL actually just picks one electrode, in that case for faces versus rest. And that electrode is a really high signal to noise. It's also the only electrode that is picked by a univariate test. For a building, it's the same thing, also the only electrode picked by a univariate test. For animals, on the contrary, there's not like a really good animal region in the ventral temporal cortex, so the model needs to use a little more noisy electrodes to be able to identify like animals, differentiate animals from rest. But the electrode with the highest contribution is also clearly selected for animals. And so we are discussing about neuroimaging a lot, and I think most of our talks are kind of geared towards fMRI, but interpretability might also be data related. And we've also seen that that data that is highly differentiable, like the subject one of Haxby, houses versus faces, can actually lead to quite stable weight maps when you look at different sparse methods. So it really depends on your signal to noise and maybe also distribution of noise, but that's something that would need to be investigated more thoroughly and mathematically, I guess. But it's just something to keep in mind that I wanted to mention. And so if we come back to MKL in general, the advantage is that it includes both anatomical and functional knowledge, but it's also data-driven. So anatomical knowledge based on the choice of the atlas, but data-driven based on which regions are going to be picked, and that's data-driven by the model. It selects a subset of predicted regions, and you can look at the contribution of each region and also at each voxel. The algorithm we've chosen is an L1 regularization, so it might not select regions with correlated information. It's actually a group lasso, so it is quite sparse. It has potential instability in the selected regions. If you run it on different folds, you might end up with a different list of regions. And it is atlas dependent. And that's also something that is fun to look at, just looking at the accuracy of your model if you choose one atlas versus another. So if you look at something that is anatomically defined or functionally defined by networks or something, then you might end up with different results. And so the last one during model estimation, and that's actually during model selection, is to include weight stability when you optimize some of the hyperparameters of your model. And these are more recent techniques, I guess, and defined in two works that I've looked at. But basically the idea is that when we optimize hyperparameters, we just look at model performance. We just look at the accuracy. But in this case, we actually will include a parameter that controls also for weight stability or interpretability based on which paper you actually are referring to. And so, yeah, I'm sorry, this is a W. This is based on the definition in that paper. But it's just a contribution of, if you take delta as the model generalization, so just the accuracy, and eta is the stability or the instability of the weight map, then you just take a linear combination of the two, and that's what you optimize, and not only model generalization. And so the kind of a main idea behind that is that there's a trade-off between delta and eta. And so if you take a little bit of model generalization, you will improve the weight stability or interpretability. And in those two works, they have assumed that you can just say it's half of generalization ability and half of interpretability, and that's what you want to optimize. I played a little bit with that, and that choice is, of course, inferencing the results. So I played with ECOG again, and that's the same subject, a slightly different task, but also faces versus what's not faces. And so we're really hoping at like pinpointing that electrode in like yellow there. And we see that if we optimize only model generalization, we obtain that map here that is not very sparse, and it gives not a super high weight to that electrode. Only 36% of the total model contribution goes to that electrode. If you do half and half, then you get higher weight, and it's 62%, and it's slightly more sparse. And if you optimize only model stability, then you get actually the weight map is quite similar, and even the contribution of that electrode is not that much different. But you see that when you fix one parameter to one and you vary the other, so I fix the contribution of generalization accuracy to 1, and I vary the contribution of stability from 0 to 4, we see that it influences the model accuracy, it influences the weight stability, and it mostly influences the map, the sparsity in the map. So overall, how your map is going to look at, how it's going to look. And of course, the definition of instability, of the weight stability or interpretability is going to affect the results, not only in how you define that, but also how you compute the distance between, basically, so to compute stability, basically you're going to compute the distance between the weight map within each fold and the average weight map across all folds. And that distance, just the way you define distance, is going to affect the results as well. And so here I took the contributions of each electrode to build that classifier. And here, instead of using the actual contribution, I use the rank of the electrodes. So I just rank them and I say, well, I mean, if the rank is always 1 for that electrode, then I assume it's a good thing. So I just use that instead of using the row contributions, and we obtain much sparser weight maps here. I'm not saying it's better, it's just that it heavily influences the results. So I think that this later version of model selection is definitely present advantages. Like it's embedded in model selection, it's data-driven, and it selects model based on 2 criterion, because if we want model stability, then it's good that we optimize on it. But I think there is quite some work that still needs to be done on those in that sense, based on first what contribution you want to give to those 2 parameters, how you define stability or interpretability, which distance you use, and also in some cases, especially when you have a really high signal-to-noise ratio, actually if you optimize one or the other, you might just end up with the same response, and so that's a lot more work, because you need to go and compute the weight map each time. And that's a lot more work for not really maybe something that's worth it. So now we're coming to like a posteriori once you have your model. And a very simple thing you can do is just average the weights within a region. So you use the same atlas, AAL again, and you just average the weight within each region defined by the atlas. It gives a list of regions, and you see directly here that this list, I use an SVM whole brain, and you see directly that this list is not sparse. That means that you need to look at the whole list. You cannot threshold the regions, the same as it goes for voxels. So I guess that it was a technique that was used a little earlier on, but it's a nice summary, but it's not really helping in any way. Then we have permutation tests, where in that case, you are using permutations. So the same way you're using permutations to assess whether your generalization ability is significant or not. You use the same permutations and you test whether the weight in a specific voxel is larger than if you change the labels and there is no information in your data about the labels. And you can either actually do the permutations or you can use an analytic approximation of the null distribution, but I think that's only for SVM. It does not apply to all the techniques. So that technique is data-driven, and it provides a thresholded map of the model that you have computed. You need to be careful when you perform multiple comparison correction. It's just a different way of doing it. If you're doing just FDR, you might be too conservative, so it might be something to keep in mind. If you are actually performing the permutations, it is pretty expensive because computing the weight maps 1,000 times is quite expensive. And then the last thing I'm going to mention, and I'm actually going to go pretty quickly over it because I think it's going to be addressed later on in the session, is that in that paper from Schrouff et al. In 2014, where they mentioned that the weight maps is a mixture of signal of interest and noise, they also offered a solution, which is to transform the weight map, or what they called a backward model, into an activation pattern that is a forward model. And you actually need to, so the goal is to kind of like take out that noise from the signal and try to identify the sources. And when you do that, you can really look at the map like it's an activation map and like it's a GLM. But it depends on the assumptions that you use to estimate the forward and the backward models, because they show different variations where they use lasso to better recover the sources, for example. But that's, again, another hypothesis, another set of parameters to set. And the activation patterns do not represent the predictive function. It's just not a predictive function. The weight map is, but that is not. So in summary, interpreting the weight maps of machine learning-based models can be complex. There are different strategies to improve interpretation. They all have advantages and limitations, whether you want something that's data-driven or anatomically or functionally driven, what kind of multivariate power you want, if you want to include stability of the patterns in your interpretation at all. Computational time, but I guess, you know, further we go, this is getting like less and less of a burden. But this is just a personal point of view. What I like to do is I look at the weight maps and I'm trying to see like, oh, maybe there is information in that region. I'm not going to say there is, but I need to go and look further into and see why this region would have a higher weight, whether it's noise, whether it's actually signal of interest. And for that, I would use other techniques, whether it's a GLM, whether it's correlation analysis. But I think they're a good starting point to see whether you have, especially if you don't have any results from a GLM and you do a classifier and you get significant classification accuracy, then they can really be a starting point to go further into your data, but they should not be taken like as the truth. This is my neural sources. So I want to thank Janina, Joao Montero, and Maria Rosa for all the work on MKL, Joseph Parvizi and his team at Stanford for the intracranial EEG data. The Pronto team, you've seen a few screenshots of Pronto, which is a software in MATLAB that we have developed to do pattern recognition for neuroimaging data. And my funding body. And there are a few references that you can go and look at. The list is quite long, and it's still growing, but you can get that from the slides and online. Thank you.